Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class, we had an introduction to RNA sequencing. We talked about different methods, for example, short read RNA sequencing, long read RNA sequencing and direct RNA sequencing. So, we will now slowly get into the methods, right. How do you actually get the data process, right. How do you set up the pipeline and what kind of tools and methods that we use. So, we will discuss this in theory and alongside we will also have some hands on. I will show you the tools in this moment, and at the end we will also have a hands on of differential gene expression analysis, where we will use these tools to actually analyze a real data set and identify genes that are differentially expressed between two samples. Okay, so, these are the concepts that we will covering in this class. So, we will talk about the RNA sequencing data processing pipeline, and in that pipeline one of the topics that we will discuss in detail is the read mapping. Okay. So, here is the pipeline for RNA sequencing data. So, we have read data in FASTQ format, we do the quality control step or QC as before, then you have this mapping and assembly process. Okay. And sometimes even after mapping you can do an assembly to do the transcriptome assembly, right. So, if we have discussed, right, if we are dealing with short read data, sometimes you would have to assemble the transcripts if you are working with these big transcripts. And after this step we do something called quantification. So, we need to quantify how many reads are mapping to each of the gene in the genome, right. So, because as we have seen that the basic principle of RNA sequencing is that the expression level of a gene is proportional to the number of reads that are mapped to that gene, right. Of course, then we need to have a lot of statistical methods, models there to actually do the final quantification. Now, we have mentioned, right, there are a lot of biases that are introduced when you actually do these RNA sequencing experiments. Okay. And to address these biases, we need to do something called a normalization, right. So, we will discuss again these normalization methods in the subsequent classes in much more detail. What it does is that it kind of accounts for these technical variations and biases that are introduced during the sample preparation, handling and next generation sequencing. Okay. So, some of these biases can be addressed computationally using different techniques. Okay. So, we will talk about these normalization methods. Again, there are different types of normalization methods, and again each method has its own advantages and drawbacks. We will again talk about that, and we will utilize some of these normalization methods for our own analysis. And once we have done the normalization, we can then do the downstream analysis, whether we are looking at isoform discovery or whether we are going to do the differential gene expression analysis. So, that is what we can do after we have done the normalization. So, again we will talk about them in the subsequent classes. So, I am just mentioning some of the tools that are available for each of these steps. Some of the tools we have already learned, and there are of course other tools that are available. So, the first one is the quality control tools, right. So, we have used already FastQC and or you can use FastX. So, similarly the quality control tool does not change because we are dealing with the same read data, right. Then we have the mapping tools for RNA-seq data, right. So, and you see these are different, right. We have top hat or top hat 2, star or high star and high set 2. Okay. So, I will briefly mention these mapping tools today. We will talk about the algorithms of one of the tools in this class. Then there are assembly tools called string tie, or you can also do something called de novo transcriptome assembly, right, without looking at the genome mapping, right. So, in that case it will be a de novo assembly, and there are tools for that as well. Once we have done this, right, once we have done the mapping and assembly, we can do quantification, and there are tools again that will help in this process. There are HT-seq or feature counts tool that actually take the mapping data and it will quantify how many reads map to each gene in the genome. There are other tools like Callisto, Salmon and cufflinks, right, that will actually analyze this data in some different way, right, and it can actually give us the quantification. Okay. So, they do not rely on the mapping data, especially Callisto and Salmon. They actually do something called the pseudo alignment. So, they give us the quantification from this pseudo alignment. So, they are not dependent on mapping. Again, we will talk about these tools when we talk about the quantification step. And finally, we will talk about the analysis step later on in much more detail, the normalization and analysis in much more detail. So, coming to quality control, we have FastQC, FastX or other tools, right. So, we have used FastQC already. So, I am not going to go into that again, right. You already know, and given any RNA-seq data, you should be able to do the FastQC check yourself now. Okay. So, we use the same parameters that we have checked before, right. Again, we have to check such as the read part base read quality, part sequence read quality, etc., adapter contamination, all those things. We check the same thing, and we do data pre-processing if it is necessary, right. So, for example, if you have adapter contamination, you want to do this trimming, or if you have some quality issues, again you want to do quality filtering, etc. So, as necessary, you have to decide on this data pre-processing steps. Again, we have discussed this in much more detail when we talked about the genome data processing, right. So, I am not going to go into that again. So, what I am going to do is I am going to discuss about read mapping. Okay. So, why is this a problem here, what is new here, right. So, we have talked about read mapping, we have talked about the tools BOTI2, right, and we have utilized that tool for read mapping in a small data set, right. So, if you remember, BOTI2 relies on Barrow-Zuller transformation, is very efficient, it does not take too much memory, right. So, what is the problem? Why can't we use just BOTI2 here? Now, one of the major challenge with RNA-seq data is that especially for higher eukaryotes or mammalian genomes, these transcripts, right, they contain multiple exons, right. So, if you look at the genes, they have multiple exons, right, and in the mRNA, right, what you will get, these exons will be joined together. You can have different combinations of exons in isoforms, and then you have the reads, right, that come from these mRNA molecules. So, if once you take the reads, right, and you want to map them back to the reference genome, what will happen is that you will get in many cases split read alignment, right. So, what is this split read alignment or gapped alignment? Is that a part of the read will be mapping to one exon, right, another part will be mapping to another exon, right. So, this is something that you need to do or need to take into account when you are mapping RNA-seq data against a reference genome. Okay. So, you have see some examples here in green, these are reads in green and orange also, right, they are mapping to this exon 3 exon 4 junction. Okay. So, when you are doing this alignment or mapping, we need to do something called gapped alignment or split alignment when mapping against the reference genome. Now, you might actually be able to solve this, right, if you are doing this alignment against reference transcriptome, but again for many organisms this reference transcriptome is not available, right, and also sometimes you might limit yourself, right, you might not be able to discover certain isoforms or variants that is not part of the reference transcriptome, right. So, in that case you also want to align against the reference genome, and in that case you would have to do the split read alignment, you would have to allow for that, right. So, that is why you need these special tools that can do this very efficiently. Okay. And there are tools that have been designed to do this kind of alignment. So, one is called TopHat or TopHat 2, this is one of the earlier tools, it is actually based on the BOTI2 program or BOTI program, right, and this actually utilized kind of this extension of this BOTI using the Barrow-Zuller transformation, and it was very efficient and fast, but now it is not used that much. Then they have the class of tools called HiSat or HiSat 2, right. So, this is something that is also quite widely used now, and you have STAR. Of course, there could be other tools, we are not going to discuss all of them, these are some examples and some tools that are very popular and we can use them as well. So, TopHat 2 I just mentioned, right, this is it did mapping using the BOTI tool, right. only an extension of BOTI. So, the same algorithm, the Barrow-Wheeler transform, and all the process is similar. If you remember the FM index, the last first mapping, all those principles they still applied, only thing it allowed is this gapped alignment. Right. So, what it did, it actually analyzed this mapping results to identify splice junction between exons, right. So, it took the same approach as BOTI2, but then it identified it looked at the mapping results very carefully and it identified the splice junctions, right. So, which exons are combining with which one. Currently it is not maintained anymore, right. So, this is something that will not use, and even the authors recommend to use the HiSet2 tool, okay, because HiSet2 also combines some of the features from TopN. Okay, so another tool is the star one, right. So, there are two steps in this process. So, it relies on something called seed searching, and then in the second step is a clustering, stitching and scoring, right. So, it sounds very familiar, right, to us, the seed searching, right. You are searching for a you are generating seeds and you are searching against the reference sequence, right. We have looked into different algorithms that will do that, right. And what it does is that it kind of involves something called sequential search for maximum mappable prefix, or MMP. Okay. So, how does this actually differ, and how can it incorporate this split alignment? Okay. So, here is the read, right. So, what this method does, it starts with the read, right. So, it will start from this sequence in the beginning, right, and it will try to find a part of the sequence or the seed in the reference genome, right. So, this brown is the reference genome, and you see, let us say it finds the seed somewhere here. Okay. So, once it finds a seed, right, in the reference genome, it will start extending, stitching and scoring, right. So, we talked about this before, right. So, what it means, it is trying to extending the alignment across the genome, and up to this point, let us say it finds, like up to this point here, it finds sub batch, and after that the batch falls, right, the quality of alignment falls. Okay. So, this is expected for RNA-seq data, right, because we need to have this gapped alignment, and sub regions will not be part of the read, right, because they are not part of the process step RNA. So, what it does, it then like stops the alignment, and then it starts taking a seed from this region again, right, from here, and then again starts searching in the reference genome. Okay. And let us say it finds a batch here in this part of the genome, and then it continues this process, right. And again maybe it finds that the alignment drops after this point, right, after aligning up to this point of the read, right, and then it will generate another seed from here, and they start searching again against the reference genome. Okay. So, this is where the sequential search, right. So, it is generating this prefix, right, the first part of the reads, right, it taking the remaining part of the reads, and they are taking the first part to generate seeds and actually searching against the reference sequence. Okay. So, this tool works well with quite a bit of efficiency, but of course, you remember, right, the seed searching is a quite a tedious process, right, and it might take a bit of computational time. So, we will talk about this tool HiSet and HiSet2. This is the version 2 now, right. This is used for read mapping in RNA sequencing data. Okay. So, the name actually comes from hierarchical indexing for spliced alignment of transcripts. As you can see, this is actually specifically designed for alignment of transcripts, and consideration of the spliced alignment is there, okay, in the tool itself. Okay. So, this is a tool that actually is based on extension of BWT for graphs. Okay. So, we have a Barrow-Wheeler transform, but for graphs. Okay. So, this tool relies on a graph, right, and it generates a graph, and then it takes a Barrow-Wheeler transform approach for the graphs. Okay. We will see in a moment a part of the algorithm for this tool, and it generates something very similar to this BOTI2, right, where we generated this FM index. So, instead of FM index here, it generates a graph FM index, right. So, it's for a graph. So, that is why it is called graph FM index or GFM. and it also does something called global indexing as well as local indexing along with some repeat indexes. So, it generates something called hierarchical GFM or HGFM. Okay. So, what it can do, like why is it used, is that it can actually generate this graph based data structure from the genome sequence, but on top of that it can give SNP information also encoded in the graph, right. It can produce a graph with the SNP information. So, what are these SNPs that are actually probably commonly found in population. So, it can also generate this graph and find these SNPs, plus it can also have the transcriptome in the graph, right. So, it can generate this complex index containing this genome sequence SNPs plus transcriptome. It can do so because it is based on graph instead of these substrings, right. So, it is much more flexible here. Again, it works in two steps. So, the first step is index building, right, as before we have seen in BOTI2, right. So, it is an index building is there, right. It would have to do this graph structure generation, right, and then followed by the read mapping or searching. Okay. So, what we will do now, we will very briefly look at the algorithm, of course, without going into lot of details into the implementation, and a part of it you will understand very easily because we have done the BOTI2 discussion when we are discussing the genome analysis, right. So, how does this algorithm work? So, let us take an example for reference genome, this GAG CTG from the paper that actually proposed this algorithm. So, what it does, it generates this graph from the reference sequence. Okay. So, here you have these 6 bases in the genome. So, we will have 6 nodes in the graph. So, we have G, A, G, C, T, G. Okay. And each node is connected by an arrow. So, if you traverse an arrow, you traverse a part of the reference sequence. Okay. So, if you traverse this part, you get GAG, right. If you traverse this part, you get AGCT, right. So, you traverse along the direction of the arrow and you traverse part of the reference genome. Okay. Now, what you can do is in this graph representation of the reference genome, you can also introduce these variants. Okay. So, here if you imagine you have a variant which instead of this GAG, so GAG CTG, instead of that it has this GTG CTG. Okay. So, for that case you have to traverse through this path here, right. So, GTG, not GAG, but GTG. So, what you can do, you can have a node and add appropriate arrows, so that you can traverse through this GTG path here, right, and you can also generate this variant genome. Okay. You can have another case here, for example, you have one base insertion. Okay. So, you can one base insertion A, right. So, in that genome, so you can, if you are traversing that genome, you have to traverse in this direction, right, CTAG, because of this insertion here. So, what you see, you can have this flexibility, right. You can have this deletion, you can have insertion, you can have SNP, right. You can have multi-base insertions in places as well, right. For deletion, for example, if you have this T deleted in another genome, right, that is a variant that is observed. So, you can simply have an arrow from C to G, right. So, that would be a valid path, right, because you can traverse then GAG C to G, right, and that will give you that variant genome. Okay. So, you can see these examples in this paper where actually they consider a lot of these variations, and for example, they also show how a deletion would look like in this graph. Okay, so, once you have built this graph, what you do is something called prefix doubling and pruning, and you generate something called prefix sorted graph. Okay. I am not going into the algorithm and how you generate this prefix sorted graph. This is discussed, this is actually developed in this paper, and they use this here, and they generate this prefix sorted graph, which would look something like this. Okay. Again, we have to understand the process of this generating prefix sorted graph before you actually get here. Okay. So, what you get is something like this, and you also have ranks added to these nodes. Okay. So, what are these ranks again? So, rank 1 means any substring, right, that is originating from this node, it should appear first lexicographically, right. So, so there is a lexicographical order. Again, we are coming to the Burrows-Wheeler transform, right. So, there is this lexicographic sorting. So, if you have rank 1 means this is the first substring that will come when you do lexicographic sorting. right. So, any substring that is originating from this A node, for example, AGCT, right, that is a substring. This will be the lexicographically fast substring, right, if we sort all of them. And it is actually very easy to generate this rank if you just simply look at the subsequent bases and then compare against the rest of the things, right. So, A is lexicographically fast, right. So, these A's will come first. So, you have 2 A's, 1 and 2, and among these it is easy to decide which one, right. So, you have AGC and here you have AGZ. So, now notice this Z was not part of the sequence, this has been added to the graph to denote this is the end of the sequence, right, and this is lexicographically the highest value, right. So, this will come last. So, if you have AGC and AGZ, then this one is the first one, right. So, this will get rank 1 and this will get rank 2. Then you can follow the same principle. So, you have C, right, this will get rank 3, then you have G, right, again you can look at the substrings that are generated from those nodes. So, you have GAG, GCT, right, or GZ or GT, right, and you can rank them accordingly, right, again based on the lexicographical order. And then finally, you have T and then the Z. Okay. So, this is the last one. So, what we do then next is we have a tabular representation of prefix sorted graph, okay, and which actually talks about outgoing edges and incoming edges, okay. And as you can see, they are actually sorted by this node rank, okay, from 1 to 11, and they have two columns called first and last, okay. So, this is something that is interesting, right. So, it is very similar to what we have talked about when we discussed Barrow-Zeller transform, okay. So, what is this first and last, right. So, let us consider this rank 1, right. So, this node rank 1 means this A here, right. So, this is the first node, right. So, we are simply in the first column here, we are simply writing the nodes, right. So, for the first rank, it is the outgoing edges, the first set of columns here, first two columns, they are under outgoing edges. So, we have the A here. So, the first rank is A, the second one is also A, right. Then the third one is C, right, but since it has two outgoing edges, we have to write it twice, okay. So, that is why we are writing C and C, okay. We just have one C in the graph, but because there are two outgoing edges, we have to write this twice, C C, okay. Then you have 4, 5, 6, 7, these are all G's and T's, and then finally 11, right, which is the Z here, okay. So, why do you write it twice? Because if you remember this Barrow-Zeller transform, this rotation, right. So, what it means, so so this is connected to both of these Z's here, okay. So, you have G here, you have G here. So, both of the G's, right. So, this J is connected to both of them. So, you have outgoing edges, you can imagine these outgoing edges, these are not actual ones, but because these are ends, right. So, this means we are connected to the start, right. So, this is the letter that occurs, G is the first one, right, first letter of these strings. So, this Z is connected to these two G's, okay. And so, this completes the outgoing edges part. Now, you have the incoming edges, okay, and again we have now given these node ranks 1, 2, 3. What we are doing here, so we are actually writing the corresponding last or the corresponding incoming edge for this first node, okay. So, for this node A, right, what is the incoming node, right. So, this incoming node actually comes from G here, right. So, that is why this base is G, okay. So, the last node that was visited right before this A would be this G, okay. So, G would be the node that comes before that. Similarly, for this one A, right, what is the last node? That is T, right. You can see this, right. So, before A you have T. So, that is why you have written T here, okay. And we can now continue, right, for this C node, right. Even though we have written it down twice, it is always G, right, that comes before this C, okay. And for incoming edge, we just write it once because we have just one incoming edge. In some cases, for example, this 5 here, right, this G5 here, right, you have two incoming nodes, right. You have one from A another from T, right. So, that is why you have written it down twice in T. For G6, right. So, this one here you see this we have written it down as Z, because we have talked about, right, there is a connection from Z to G which we imagine, okay. And we can complete this table along this way, right. So, we have completed this, right, and what you get is this last first mapping property, okay. And you remember last first mapping, right. So, the ith occurrence of a letter in the first column and the ith occurrence of that letter in the last column, they correspond to the same letter in the reference string, okay. So, we talked about this in biocellular transformation. So, this applies here, right. So, this G correspond to this G because of this last first mapping property, okay. So, what you can do now, we can utilize this last first mapping property in this table, and we can map certain reads, okay. And what you see here, we have seen is that it is very flexible, right. You can actually accommodate mutations, you can accommodate these splice variants, right, because of these graphs, right, because of you have converted this reference into a graph, okay. So, we have this global plus local indexes plus some repeat index which gives us this HGFM. Okay, so, how do you search for a substring now? Okay. So, let us take this example, we have GCTG, right. So, we can start from the last one, which is the G, and see where these G's are, right. So, these G's are here, their correspondence are given by these red arrows, and we can go to the last column, right. So, which one is the letter that occurs before G, right. Remember, this is the principle that we have in the last first mapping, right. So, the T is the letter that occurs before G, right, and we have found T here in two cases. So, we need to follow up on those two paths, and this T, because of last first mapping, they correspond to these two T's here in the first column, right, and only for one of them we find C, right. We are looking for C. So, we can find C, and this C corresponds to that one there, right. This is the second C, and then we have this G. Okay. So, we have found the substring GCTG. Okay. So, it follows the same way that we have discussed before, right, and we can find any substring that we want. Okay. So, what you can now imagine, right, we can extend this graph to generate graph with splice variants, and we can do split read alignment, right, because of this flexibility that is built into this algorithm. Okay. So, these are the references that we have used. So, what I wanted to do now is I just wanted to show you the HiSet2 program, right, before we actually conclude for this class. Okay. So, I will go to this HiSet2 program now. Okay. So, let us see, right. Okay. So, here is the website where you can actually get this program from, and you have the manual, you have download options on the right hand side, right. You can see the download option, and you can download any version, right, of this you want. And also what you will see, you have some of the index files that are already built and available. Okay. So, here are the versions of for you have for OS X or for Linux, right. So, you can download any version that you appropriate for your system, operating system. So, in this system we are running WSL. So, we can use the Linux version, and we can use any of these binaries, right, any of these versions, right, of HiSet2. In addition, you will see some index files that have already been built. Okay. So, this is very useful, right, you do not, so which means you can skip the first step. If you are working on any of these organisms and you have this index file available, you can skip the first step when you are running this program, right. So, you have this already given genome plus transcriptome, right. So, you have this here genome SNP transcriptome, right. So, where you have the SNP data already and part of the index. Okay. So, if you have these files available, you can simply download them and you use these index files, and you can do the mapping very easily. Okay. And you have the manual here, this kind of describes how this method is developed, right, and also what is the running mode, etc., alignment summary that you will get, etc. Okay. So, what I will do is actually I will run it from the command line. So, I have actually downloaded this program. So, here so we can go to this directory cd RNA-seq data analysis, right, and inside this we can see this HiSet2. So, you will probably notice this here, it is highlighted already, right. This is the program that I have downloaded and extracted, okay. And I can simply go inside after extraction, I can go and call this program, okay. One of the things you might want to check whether you have permission to run, otherwise you can change permission through chmod, okay. So, this is very simple actually. Once you download the program, you can simply access, and you can also say -help. You can simply call this from here, or you can add to path, right. In the last hands-on we discussed how you can add this program to path. You can do that as well, so that you can simply call HiSet2 and it will be available everywhere in the system, okay. So, I will just simply run HiSet2 -help, okay, and you will see certain options here, okay. And what you will notice, right, is the usage is very similar to the program that we use BOTI2, right. So, it is very similar to what we have used earlier, okay. So, HiSet2 -x HT2 index. So, this is the base name of the index file. Then you have the input file options, right, -1 M1 -2 M2 or unpaired data, right, or if you want to give SRS accession number, this is from a database, it can directly run that accession number, and the output in a SAM format, right. So, this is very good, right, because we already have learned all this before with BOTI2. So, we can simply follow that and do the same thing here, okay. So, there are few extra things you will probably notice. So, when you come down to these options, right. So, the options again they look very similar to what we have discussed before, right. So, you have this FRET33 etc., all these options, trim 5, trim 3 etc., fast, sensitive, very sensitive, these options again, right. So, these kind of things are there, but along with that what you will see now is another set of options for spliced alignment, okay. So, you can see this, right, whether you want novel splice site out file, right, or if you say no spliced alignment, you do not want this spliced alignment because you are you are mapping to reference transcriptome. So, you do not need this. So, you can also have these options. So, these options are actually extra compared to BOTI2. And then finally, the output is also in SAM file, right, and which means we know the SAM format, right, and we can interpret the data very easily. Okay, so, we will use these tools, right, later on when we do the hands-on. I will demonstrate further and we can go back now to the conclusion. Okay, so, to conclude, right, so we have discussed about the read mapping step and some of the algorithms that are used for this process, and this is one of the first critical steps of RNA sequencing data processing pipeline. The challenge we have discussed, because we in many cases we need this split read mapping, right, if we are aligning against reference genome. So, we cannot just simply use BOTI2, we need some sort of other modifications there in that algorithm. We talked about the star and HiSet2 a bit more detail, and we have seen that HiSet2 uses a BWT for graphs, right. So, Barrow-Wheeler transfer for graphs, and it creates a very similar HGFF index for read mapping, okay, and it utilizes this HGFF index to actually search for splice variants and the reads in the reference genome data. Thank you.